Thank Dave and everybody for the invitation to come and speak today, and thank you for that very nice introduction. I am not, in fact, going to talk about molecular level lipidomics. I am not going to talk about radiocarbon. I'm not going to talk about anything he just mentioned. And in fact, what I'm going to talk about mostly today is nitrogen, and so it's a real shame that Angie had to leave. So Angie Knapp, who's been here visiting, provided, in fact, a lot of some of the background advice for helping us get started in what I'm about to talk about. So just a plug for Angie, who was just here. A lot of the credit of tips of how to make this all work really does go to her. So I'm going to do what people often do, which is to basically stitch together two different subjects, two different talks, and hopefully you'll see that there's at least a little bit of commonality or connection between them. So first we'll start talking about nitrogen and talking about a problem in oceanography in sort of deeper time, and then we'll start talking about something completely different. I'll talk a little bit about archaea and their role in the ocean. So, oh, it works. That's great. This is my first attempt at 21st century
we should make a statement
technology.
That she came here with no
computer.
in hand. She came here and she put it up on Dropbox. She brought it up on Dropbox.
And there it is.
Here we are right now. So
sorry, I didn't bring my computer.
Forward.
It's the first time I've ever done that. So, and it works. It's easy. So I'm never taking my computer anywhere. Yeah. So I'm interested, more broadly speaking, in what happens in marine biogeochemical cycles at the level of biological production, how that works in terms of the chemical signatures that the entire community productivity in the ocean generates, and how that translates to sediments in order to give us longer-term sedimentary records. And so while I'm not a practicing paleoceanographer, I do view a lot of what we're doing from the viewpoint of trying to understand how well sediment paleo records preserve signals with reasonable fidelity, because there's incredible complexity, as we all know, between what happens in the sea surface, the biological productivity that then is chewed up through both larger scale food web and also through microbial biogeochemical processing, how everything gets processed and mixed through the biological pump. Eventually some small fraction of it is sedimented, and we're left with the dregs and the residues of all of this great and wonderful complexity by the time it gets buried down here. And somehow we trust the idea that all of that stuff that's happening in the water column is in some way proportionally translated into the sedimentary record. And of course we know that when we look at sedimentary paleo records, we can have decent metrics that allow us to believe that time is accumulating in a vertical fashion. We can trust through a variety of dating techniques, and sometimes, if we're lucky, to really obvious visual techniques, that these sediments have a history or fidelity of recording the paleoclimatic and biogeochemical history of what's happened in the overlying water column. And yet you can imagine that it's really quite remarkable that all of these various records do give us some coherent signal over time, considering the craziness of what happens to that organic matter between where the majority of it is produced in the photic zone of the surface ocean and all of the intense reworking and processing, heterotrophic respiration, and all the other massive amount of enzymatic destruction, reworking, and everything that happens to it as it descends the water column towards the sediments. And so this is a very, very famous Martin curve, and where John Martin and colleagues went around, there's a lot of these biogeochemical flux studies like JGOFs and some of the other things from the 70s and 80s, went and determined the power law or exponential decay functions for the loss of organic matter in the water column, showing that upwards of 90, 95, 99% of the total organic matter produced in the surface ocean is destroyed, and all the rest of it is really strongly reworked by the time it gets to sediments. And so I'm really interested in whether we can believe that what we find down here at the bottom is a record of what happened up here at the top, with any kind of systematic ability to trust that that's true. So the premise that I'm going to operate from is that this is true, and hopefully the two examples that I give you today will go some way towards convincing you, based on evidence, that this is true. Is that despite the complexities of what's happening in the biological pump and all of this huge remineralization of carbon in the water column, we do still manage to integrate the full sum of processes that are happening throughout the water column, and that these records do get transmitted to sediments and preserved with a reasonable amount of fidelity. Okay, so the two things I'm going to talk about then. The first example is the one about nitrogen. So this is about Cretaceous Ocean Anoxic Event 2. So I'll give a little introduction to that and tell you what we learned about that. And then I'm going to transition and talk about archaea, lipids of archaea, and how we use those as a paleotemperature proxy, or don't use them, depending on your point of view. So to go back to the Cretaceous, I don't know if anybody here works in the Cretaceous or has ever been to see this particular formation, but about 90 million years ago, there was a massive episode that is thought to be globally distributed, but certainly was distributed in the developing Atlantic and the closing Tethys, of ocean anoxia accompanied by deposition of a very thick layer of incredibly organic-rich sediments. And so this is known as OAE2. This sediment thickness here is about 1 meter. It's just a little bit more than 1 meter. This is an expression that is now currently, obviously, uplifted out of the sea. This is in Italy. So this is the particular horizon known as this is the Bonarelli horizon. It's known as the Bonarelli event. And this is the best on-land expression of this thing, is this very famous outcrop here at the town of Furla. And I was able to go see it, and it's just absolutely spectacular. Because this is completely bright white organic poor limestone with a few interbedded cherts here and there. And then there's this meter of organic rich black shale that's up to 20% organic carbon, and then it immediately transitions back to this limestone and goes for another several, so it's kilometers and kilometers on either side of this horizon. So the question is, what the heck happened here, and what is this telling us about productivity in the ocean? And at the same time, does this record really tell us about productivity in the ocean, or is it some diagenetic or dissolution event that happened after the fact? And so we think we can go after this and answer both questions. The work here was all done by my PhD student Maytel Higgins, who is now at Princeton, which is where Angie Knapp formerly was, which is how we have all these connections that allowed us to go down this road. So while she was a student, she had 2 children also. So, really a remarkable person. Okay, so here are the 2 things that have been debated in the literature to explain OAE2 over the years. One hypothesis is that what you see here with this carbonate-free, organic-rich horizon is that it is the relic of a more or less stagnant system that was incredibly stratified. The ocean stratification led to depletion of oxygen in bottom waters, and this oxygen depletion then led to extremely high preservation of organic matter, and such stratification is associated with very oligotrophic systems, and so this is a cyanobacterial ocean in which most of the productivity is nitrogen-fixing cyanobacteria, because the surface waters are nutrient poor, so they're able to be the ones who make a living. They don't produce a huge amount of biomass, but that biomass sinks and it's almost quantitatively preserved because the deep ocean is so anoxic. This is a reasonable explanation. The alternative hypothesis is also a reasonable explanation, which is that there's something happened globally that caused the nutrient balance in these particular regions of the ocean to be disturbed in some way, that caused eutrophication and overproduction. In other words, there was an extra influx of nutrients, either from land, from some kind of intensified upwelling circulation, or some combination of the two. And so this is really what we're after. Is this a eukaryotic high productivity event, or is this a bacterial low productivity but high stratification event? And so basically these are the 2 options we're after. Do we have a blue water system, or do we have something that was just growing like crazy? Okay, so our solution to tackling this problem was to go straight after the idea that nutrients can help us explain this. Because if we have a system that is nutrient poor, we should be able to tell that apart from a system that is nutrient rich, based on the signatures of nitrogen isotopes that are preserved in this organic matter. And nitrogen isotopes in the marine system are very complicated, and thankfully we're going to ignore most of the fractionations and reactions that are on this particular slide. I just put this up here to show you that there are a lot of routes and exits for nitrogen into and out of the ocean and sediments, with a lot of complicated transformations that occur throughout. And so this is potentially a very complicated process in order to try to interpret values of nitrogen isotopes or nitrogen isotope fractionation. But one of the most important fractionations associated with everything on this entire figure here is the fixation of nitrogen into organic matter by nitrogen-fixing cyanobacteria. So nitrogen enters the ocean and is fixed in organic matter with effectively no fractionation. And this becomes really important. So if your ecosystem in the photic zone is nitrogen depleted, and is trying to replenish its deficit of nitrogen by fixing nitrogen to make up that difference, that nitrogen is going to be fixed with the delta N15 value of near 0. And this is a pretty good signature for nitrogen fixation because most biomass is made from nitrate, and nitrate in the ocean has a value on average, at least in the present day, of around +5 or +6 per mil or so. So that gives us sort of a 5 to 6 per mil end member type signal that we might be able to look for. Historically, looking at nitrogen isotopes during this event has led down the road of suggesting that delta N15 values from this Cretaceous OAE2 suggest that indeed we were looking at 100% nitrogen fixation in surface waters. In other words, we were looking at the cyanobacterial end member option. And the reason for that is if you measure the bulk delta N15 value of organic matter, you see the values are very low. At the same time, there's another index of something for certain biomarkers known as 2-methylhopanoids. The hopanoids are bacterial biomarkers, and in particular, these bacterial biomarkers with a methyl group, what we call the 2 position, are made by some types of cyanobacteria that fix nitrogen. And so the idea is if this index of 2-methylhopanoid, or the percentage of 2-methylhopanoid is high, there may have been a lot of nitrogen-fixing cyanobacteria. And so when you see a high index and low N15 values occurring together, a reasonable conclusion is that there was a lot of cyanobacterial production. There is a bit of a problem with this, though, which is that, first of all, depending on where you go, the delta N15 value during this event is different. And the second is that in some of those places where it's different, the N15 value that you get through some of these characteristic sections is considerably lower than 0. And the real practical limit for the minimum value of nitrogen-fixing cyanobacteria is no less than about -1.5 per mil. And in fact, it may only be about -1 per mil. Because the fractionation is somewhere between 0 and 2 per mil, but the dissolution of atmospheric N2 between the atmosphere and the surface ocean, that also has an equilibrium fractionation on gas exchange, which makes the N2 in the surface ocean heavier than 0. And so if you put a 0.5 to 1 per mil positive fractionation on dissolution of the gas and then subtract a 0 to 2 per mil fractionation on the fixation, you can't get to values that are -2 to -3 per mil in your biomass. And throughout the Bonarelli and also throughout the black church that predate the Bonarelli, all these values here are around -3. And so it becomes very difficult to figure out how this data are compatible with an N fixation signature. So we were very curious about this. It's one of the things that helped us to get started on this problem, was wondering if there's more going on here than meets the eye. So the thing that Maytal wanted to go after, and I have to give her a lot of credit for this, because she came to me and said that she wanted to do this for her PhD project, and initially I said, no way. This is so risky that you don't want to set yourself up for something that could potentially fail like this as a PhD level project. But she convinced me that this was worth doing, and she was right. So she was braver than I was. Her idea was that she would go back to some of the classic literature on trying to look at nitrogen-15 isotopes in porphyrins. Porphyrins are the degradation products of chlorophylls, and use that specifically as a way to look at the N15 values of biomass in surface waters. That way it wouldn't be compromised by diagenesis in the sediments or degradation down the water column, because by definition, a photopigment is only going to be made by organisms living in surface waters. And that way she would at least be able to isolate what the nitrogen sources were to the photosynthetic fraction of the total biomass. And we could then compare those values to the total nitrogen in sediments to see if somehow the sediment value was not reporting what the true primary producers were using. So chlorophyll is a tetraperol with a phytyl side chain on it. It has 4 nitrogen atoms, and no matter what happens to it through breakdown and diagenesis, as long as you preserve that tetraperol ring, those 4 nitrogens are the same nitrogens as were originally made by that organism at the time it grew. And so there's no opportunity here for fractionation of those nitrogen isotopes. If the molecule breaks down, the porphyrin ring is busted up and it goes away. And so you don't have any more of that in your sample. So you have intact nitrogen and all the side chain modifications that happen during diagenesis, and there are dozens of them. There can be hundreds of different porphyrins in the sediment. As long as you have the ring, you have the original nitrogens. And so we were not interested in characterizing the diversity of structures that we found in the sediments, but rather in getting the total porphyrin content and using that to measure a delta N15 value. And so what she did was she came up with a multiple-step HPLC fractionation method that was designed to first separate out the individual porphyrins, and then in an extra dimension to clean up any residual nitrogen-containing compounds, to put them back through and compact them together in a very narrow elution window. So this is backwards from what you would normally do in chromatography. Normally you do a crude cleanup first, and then you do an individual compound separation to characterize everything. We did it the other way around, because this way, our very last step collects material across a minimum of time, and this minimizes our background contamination issues. And so we were able to get our background nitrogen contamination, which organic material is very, very prone to, down to very, very small numbers. So doing this, she was able to measure N15 values on a variety of different test samples. We did lots of controls, things like that. And the other nice thing about working with porphyrins is that when you do get them in purified aliquots, they have just wonderful, beautiful colors. All of these are porphyrin samples, and their colors depend on which metal is conjugated to them, and also how many double bonds they've lost in the process of diagenesis. So it was really spectacular to look at. The next thing that we realized we needed to know, other than figuring out how to make the measurement, was that when you measure N15 values in chlorophyll, that's not the same thing as the whole cell. And the whole cell doesn't have the same value as the original nutrient that it was using as a substrate. And what we're really interested in is what nutrient was the cell using as a substrate. And so we needed more work to try to figure out how to go from this number to correct for the biosynthetic fractionation, and then see if there was some way we could figure out the difference between cell and nutrient. Other people have worked on this problem a lot, so these numbers are fairly well known. But this fractionation, or this isotope effect here, is not very well known. So the idea is, of course, that if you are going to measure your N15 record in the sediment, so this is downpour in the sediment, so depth or time, here's your delta value, and this is just some simulated record. And so if we're looking for wiggles in a record of a pigment, we want to know what the original biomass was, and so we hope that we can understand the offset between the pigment and the biomass. And so we call this epsilon, or isotope effect, for synthesis of porphyrin. And it's just defined in the approximation of epsilon is equal to subtracting delta values. It's just defined as the biomass value minus the chlorophyll value. And so if this were a normal biosynthetic reaction, your chlorophyll would be isotopically negative relative to your biomass, and so this minus a negative becomes a positive number. Okay, so a regular fractionation is a positive number. In eukaryotes, this difference is constant 5 per mil. In other words, if your biomass were 0, your chlorophyll or porphyrin would be minus 5 per mil. And there is a lot of literature on this about this fractionation in eukaryotic algae. Julian Sachs, who's now at University of Washington, did this for his PhD work when he was a student in the Woods Hole Joint Program back in the mid-1990s. And what Julian did was he grew a variety of eukaryotic algae, and he measured species by species, culture by culture, this value of epsilon porphyrin. And so 5 per mil is the eukaryotic average of all cells, and the standard deviation on this is plus or minus 1 or 2 per mil or so. And this has been the basis then for interpreting values of porphyrin N15 throughout the literature. The idea that chlorophyll is 5 per mil lighter than associated biomass. What's interesting about this is that there's one paper in the literature about a cyanobacterium, Anabina. So this is a heterocystis nitrogen-fixing cyanobacterium that grows in fresh water. And this was published by Beaumont and Janke, Linda Janke from NASA, who measured chlorophyll from the cyanobacterium and found that it was almost 10 per mil isotopically heavier than its associated biomass. And this had just been sort of put on the back burner, and it's the only measurement out there. And so nobody really knew what to say about it. Is it a mistake? Is it, do we have any idea what's going on? So Maytal's first major project was to go and see if she could add more data to the database of this chlorophyll fractionation. So on Julian Sachs's algae list, he only had what we call heterocont algae. He didn't have any primary or basic green algae or primary or basic red algae. So she did some of those, and they, again, all have these epsilon pore values around 5 to 6 per mil, or chlorophyll 5 to 6 per mil lighter than biomass. We grew them in a variety of different ways, with different nutrients, with different light-dark cycles, with a variety of culture conditions. And then she grew a whole bunch of cyanobacteria. And all of the freshwater cyanobacteria all had these weird Anabina effect values for the fractionation of chlorophyll relative to biomass. They all had, on average, chlorophyll that was 10 per mil isotopically heavier than the associated biomass. This is a huge fractionation in the wrong direction. And it didn't matter whether we grew them as nitrogen fixers, or grew them on ammonia, or grew them on nitrate, or grew them on anything. The nutrient supply made no difference at all. And then we grew some marine species, Synechococcus, a marine Synechococcus, Crocosphera, Prochlorococcus, and these on average are 0. And so they're basically, they give chlorophyll values that are the same as the biomass from which that chlorophyll is extracted. And so we ended up with this very odd, taxonomically specific, or maybe specific, we don't really know, we still don't really know how to explain it, but we ended up with this very odd taxonomic sorting of the data, in which if you had a system that was dominated by eukaryotic algae, the biomass would have a particular N15 value and the chlorophyll would have a value that was lighter. If you had a system that was dominated by marine cyanobacterial growth, you'd have biomass at a certain N15 value and chlorophyll that was at the same value. And if you had a freshwater system, biomass would have a certain value and the chlorophyll would be heavier. And so we were interested in using this as basically an indexing way to get at what kind of biomass is in the sediment. And this was completely unexpected. We had no idea that this was going to happen with the data. It became more useful to us to look at this difference than it did to look at the absolute value. So now our question is a little bit changed. Instead of just saying, okay, we're going to look at the absolute values and how they change through the OAE, now we're really interested in whether these lines become more close, well, they're parallel, but whether they get closer together or whether they get farther away during the event. So Maytal was able to get some of this sediment from Demerara Rise, from one of the ODP cores. This core had previously been published for a C13 isotopic record, which defines the isotope excursion, defines the anoxic events. And we put some nitrogen bulk values on that, so we measure nitrogen in the bulk organic matter, both in the total nitrogen and in a demineralized kerogen fraction, just to check it. And then she measured the nitrogen isotopes and the porphyrins. Among the other interesting things that we didn't elaborate on too much is that the timing of this minimum in the nitrogen isotopes is at the end of the OAE, whereas the carbon isotopes start going up at the beginning and last throughout the OAE. That's pretty interesting. But in the end, what we were really after was calculating this value of epsilon porphyrin. So the epsilon porphyrin in this core is staying in the area of 4 to 5 per mil. So to look at it in a slightly bigger version here, if we had 0 epsilon porphyrin, so chlorophyll and biomass the same, we might expect that that was the equivalent to 100% of the export biomass from diazotrophy. So we have basically a cyanobacterial population. If we had it at 5 per mil, then we would expect that there's no diazotrophic export production. And our values sit at 4 on average, 4 plus or minus 1. And so we think that this points to about a 20% contribution of cyanobacterial biomass to the export production, which would mean that the event is dominantly a eukaryotic overgrowth, a nutrient-rich, high-productivity system. And so that is our interpretation that we put on that. What's interesting is that this is more cyanobacteria than we currently have today in most open ocean environments. It's at least double, if not triple, the cyanobacterial productivity. So this is still compatible with a lot of the biomarker records that are already out there that suggest that cyanobacteria were abundant, at least based on biomarker ratios. It's just that they were abundant because they were making up for nitrogen deficits, but the overall system was more productive. So since we published that, we've been working a little bit with Andy Ridgewell and colleagues in Bristol, and they are modeling the nitrogen speciation in the ocean and trying to overlay that on their productivity model for Cretaceous OAE2, which they've already, I think, now published in Paleoceanography, maybe earlier this year, late last year, suggesting that there was 3 times as much phosphorus in the ocean during OAE2, and that it indeed was an overproductivity event, and that was what was driving it. And so our results would agree with what they say. So this points to a really different kind of ocean system in the past than we have today. So today our ocean is oxidized down into the deep waters, and as a result the nitrogen budget of the ocean is dominated by nitrate. Most of the nitrogen in the ocean is nitrate. So oxidation of biomass nitrogen, which is reduced nitrogen, proceeds vigorously throughout the ocean, and that nitrogen, as the biomass is degraded and spit out as ammonia and reduced nitrogen, is oxidized through nitrite and nitrate, and then upwells into surface water to drive production, mostly in the form of nitrate. The previous hypothesis about a heavily nitrogen-fixing sort of cyanobacterial ocean in OAE2 would suggest there was a nitrogen-deficient upwelling flux. So you might have had reducing deep waters, a lot of denitrification, pushing a lot of nitrogen deficiency into surface waters, promoting the growth of cyanobacteria. But instead, we suggest that basically this anoxic or reducing deep ocean penetrated up into the base of the photic zone or into significant regions of local upwelling, such that there was actually an upwelling ammonia. And we have an isotope model that I don't really have time to talk about here. We have an isotope model that is possible to reconcile bulk delta N15 values of -3 and -2 per mil if there is an ammonia flux directly into the surface ocean through upwelling, which would indicate that in these local regions where we have this intense expression of the OAE, what we had was reducing conditions that were strong enough to allow persistence of ammonia through the upwelling zone because the rates of nitrification were not quite fast enough. So this is interesting, and I hope that we can explore this.
From the paleogeography, is there any coastal to open?
There is actually, yeah. So we can look maybe later, but I think that, yeah, you know, I don't know if that's in the follow-up slides. It might be. So when we run the Gini model on this, we get the negative values are at the most redox, unstable boundary zones. So somewhere where it's incredibly anoxic, you don't get a lot of denitrification, and you don't get a lot of nitrification, and so you don't get a lot of fractionation of any kind, and so you don't have these weird isotope values. But on the boundaries of those regions, you get both denitrification, nitrogen fixation, and ammonia upwelling, and the balance of those are at least isotopic branch points, and that's what allows you to have negative values. And then when you get into fully oxidizing ocean, then everything's nitrogen. So yes, so it's negative in England and Italy and in the northern boundary of the Tethys and the widest opening part of the Atlantic, and everywhere where it's narrower, it's not as negative. It's really cool. Okay. So, part 2. So the transition then between that subject and this subject is that the process of taking reduced nitrogen from the remineralization of organic matter that is degrading in the water column and taking that ammonia and organic N and oxidizing it back to nitrate and then eventually to nitrate, for a long time that was believed to be done by nitrifying bacteria in the ocean, but these days we're now pretty confident that nearly all of that is being done by nitrifying archaea. That these archaea are the major players in nitrification in the deep ocean. And so they are responsible for the fact that when you go out and you measure your seawater, all that nitrogen out there is in the form of nitrate. Although technically they only take it as far as nitrate. So somebody else is responsible for the nitrate. So many or most of you are familiar with this at some level, but in case there's anybody who isn't, what we're interested in talking about is how these archaea are used as records, as a new organic paleo-sea surface temperature proxy. These archaea are distributed widely throughout the global ocean, and they're called Marine Group 1 archaea. They were called for many years Marine Group 1 Crane-Archaeota, but they're now classified as something called Thaumarchaeota, which is a more basal phylum of archaea. And they make, as their membrane lipids, a complex series of structures that we call tetraether lipids or isoprenoidal tetraether lipids. And the abbreviation for this is GDGT, or glycerol dialkyl glycerol tetraether, which nobody remembers, and so everybody just calls it GDGT. And these GDGTs, throughout their structures, are all based on this same backbone of a parent structure, but sometimes they have cyclopentane rings down the chain, and they can vary the number of cyclopentane rings. And so we call them by abbreviations like GDGT0. This is GDGT0, no rings. This is GDGT1, one ring, etc. And similar to the alkinone paleotemperature proxy, colleagues from the Netherlands, primarily the group from the Royal Netherlands Institute of Sea Research, otherwise known as NEOS, went around and compiled ratios So this should be sums, sorry, this is GDGT2 plus GDGT3 plus archaeal. And compiled ratios of these compounds and looked for something that had a linear behavior relative to sea surface temperature. And so this is an empirical calibration. And the idea behind it is that when an organism needs to have more flexible membrane lipids, I'm going to get this backwards now. So an organism is going to have more of the GDGT. Oh, Amelia, help me. I got it backwards now. Yeah, more of the higher ring numbers when it's colder. Yeah, so for more flexibility. And so you can go through sediments everywhere in the surface ocean and measure a profile of these lipids and count them up by the number of rings. And this ring index goes up as the temperature gets hotter. And it goes down as the temperature gets colder. And then you just calibrate this with a line, and then you can go around and make sure it works everywhere by doing a core top calibration. And then you can go backwards in paleo records and reconstruct the sea surface temperature in the past by looking at this characteristic ratio. So this works really well. In fact, I used to have a slide in here about Jessica Tierney's recent paper from Nature Geoscience from a couple of years ago, in which they were able to reconstruct the recent, so just Holocene and modern day temperature rise in East African rift lakes using this index, and find that it was just absolutely accurate in terms of timing and sensitivity. You can give just a couple degree wiggles in temperature that perfectly match what we know about temperature records. And so it gives a very nice relationship to temperature and climate, and the records are great. But these lipids are not directly recording sea surface temperature. This calibration of this ratio to the overlying sea surface temperature is based on core tops. So you go around and take core tops, measure the ratio on the core top, relate it to the overlying sea surface temperature. It makes a great linear relationship, and use that relationship back in time. But that doesn't require that that calibration be a direct function of the sea surface temperature. And so what I'm going to do is talk about what we know about where these lipids actually come from, and then ask questions about where we might go in the future in terms of being able to try to figure out why it's correlated to sea surface temperature. So I'm going to show some data that basically establish the following things. That where nitrification happens in the water column, the C13 values of the GDGTs, the radiocarbon content of the GDGTs, something that we're calling GDGT 2 to 3 ratio. This comes from Rich Pancos and his group in Bristol. All of these things point directly to the idea that GDGTs come from the base of the photic zone and below. So they are subsurface components that are then exported to marine sediments. So why do we think we know this? The number one reason we think we know this is because now that there is one of these archaea in culture, it is an autotroph that lives by oxidizing ammonia. In other words, it's a nitrifier. So these guys are the major agents of nitrification in the ocean. And we all know that most nitrification happens below the photic zone. So organic matter is remineralized, and the zone of remineralization is where the maximum rates of nitrification happen. In fact, you can go out and you can enumerate either cells of these archaea, or you can enumerate RNA transcripts for archaeal ammonium monooxygenase. You get the same answer approximately either way you do that. And you can look down the water column and find that they are low in surface waters. They start to increase right at the base of the photic zone. They max out by about 200 to 250 meters, and then the activity then decreases as you go into the mesopelagic. And there are a few such records like this, and they all basically look the same way. And they're correlated with the slope of the rise in nitrate in any given location. And so these archaea, they live, they're active, and they're producing the fact that your nitrate profile would go up at approximately this exact same depth and slope. The interpretation in the past, or at least the justification for saying that a Tex86 archaeolipid ratio that makes it to the sediments is a tracer for surface water, is the idea that it's these cells in the surface water that are packaged preferentially through grazing and particle sinking. So the particle flux is coming from here, and so you're not getting these lipids. So you're just packaging up these archaea, even though they're a small fraction of the total, and exporting them to sediments. Oops. We know this isn't true for a couple of reasons. I think I can just skip that and go back to it for a second. One of the reasons that we know this isn't true is that if you measure the radiocarbon content of these lipids in sediments and you compare them locally to the planktonic foraminifera that occur in the exact same sediments, planktonic foraminifera record the surface water because that's where they're calcifying and that's where they sink from. And if you measure the GDGT lipids in those same samples, they have less radiocarbon. And so this is a downcore sediment record from Santa Monica Basin from my student Suni Shah, and she has benthic forams, planktonic forams on a very high sedimentation rate and fine scale in this core such that we record this change here, which is the bomb radiocarbon pulse. So due to anthropogenic nuclear weapons testing in the early to mid-1900s, 1950s, 1960s, sea surface water radiocarbon in DIC has gone up, and now it's decaying back down to a neutral background. And so there is this timing pulse that gives us an accurate sedimentation rate, an accurate date for the sediment, but it also usefully gives us an accurate change in the isotopic composition of surface water DIC. And so we can measure the planktonic forams that change, but the bomb radiocarbon has not penetrated really significantly to the deep, so the benthics are more or less a straight line. And the GEGTs are right here in the middle. And so, and we measured several different compounds at several different depths, and earlier, so this is 2008, in 2001 I published the values from surface water that also fall right in this box as well. And so we have several data points, a bunch of data that basically shows that these things don't come from surface, they come from deeper in the water column. The next thing that you can do is you can say, well, does anything else agree with that? One of the things that is interesting about thinking about how much archaeal production occurs in the water column is that we could make assumptions about the cell count per volume or the growth rate per volume as a function of AMOA transcripts. You can do this either way again. And you can calculate a presumed lipid production rate, and then you can calculate the integral under this curve. And you can basically say, here's my production here, and integrate everything, and integrate it through a maximum export threshold of something like 1,000 meters or 1,500 meters, beyond which you assume that nothing else is happening. And you can do that and come up with a curve that looks something like this, where in addition to integrating the predicted production, you can also impose a range of degradation constants, rate constants K, for lipid degradation rates in the water column. These would be drawn directly from things like the Martin curve, or actually in our case more directly from some of the work of Stu Wakeham. And so you can impose a really broad range of reasonable Ks that have been found for a breakdown of various kinds of lipids. And you can impose that on the integral sum. And so for every horizon, you can sum up production, and you can apply exponential decay function. And by doing that, you can end up with two estimates. One, you can end up with an estimate of the maximum and minimum average temperature of production of the total lipid content of the total water column. And that's what this purple line is. Your purple line suggests that if you have a very fast degradation, you could end up with temperatures as low as plus 3 degrees, and if you have slow degradation, you could end up with something as warm as about 12 degrees. But in the location that we're modeling, the sea surface temperature is 25 degrees. And so no matter what you do, even if you degrade lipids quickly, degrade them slowly, if you have a situation where you're producing most of your biomass below the photic zone, it's really hard to figure out how you could subsequently go backwards and integrate that biomass to have most of the flux reaching the sediment only be from the 25-degree surface water. Similarly, using that exact same function, you can predict the delta C13 value of those lipids and compare that to what's known in terms of the measured delta C13 value of those lipids. And the global average value for delta C13 for GGTs is minus 20 per mil. And minus 20 per mil falls right in the middle of our rate constants curve right here. And it suggests that, again, minus 20 per mil means that most of that biomass comes from this sort of rate constant, or comes from about 8, 9, 10 degrees or so, which is again consistent with an average export depth of something like 200 meters or 250 meters in the water column. On the other hand, if everything were exported from surface waters, the predicted delta C13 value of these lipids would be minus 19, or a whole per mil unit heavier than what the actual observation is. And the reason we know that is because this fractionation between DIC and lipid has been precisely measured to be it's actually about 19.8 per mil, plus or minus a couple tenths, so basically 20 per mil. And so when your surface water is +1 or so, your lipids should be about -19. And so there isn't any way you can produce lipids that are this light unless your average DIC at the depth of production is more like 0 per mole. And so you must be within the chemocline where these lipids are being produced. And so we would explain this through the following, to basically say that the normal argument that your lipids are exported from the surface water, because even though there aren't many of them, they're more prone to being packaged and sinking, is counteracted by, basically the argument is that if you have deep water production, they're not really incorporated into particles, and that's probably true. So the probability that any individual archaean is actually going to be a particle incorporated down at this depth is probably much lower than in the surface. But there's so many more of them that it basically outweighs the first effect. Plus, the deeper it is in the water column, the more it's bypassed the zone of intense mineralization, and so its probability of decreasing its concentration further or degrading further from that point actually goes down, because it's spending less time in the water column in a less intense zone of remineralization. And so, yes, you do get lipids from throughout the whole water column, but the bulk of them come from about right here. The other, probably, I think maybe the best thing that points to this is the work of Rich Pancoast. Rich and his students have come up with something that is just a really astute observation, which is that he noticed that there is a variation in the ratio of GDGTs 2 and 3. And I don't know why this popped out at him, but it was a great observation, which is that basically, if you look at profiles, you can look at GDGT2 and GDGT3, and in surface waters, these compounds are of approximately equal ratio. But if you were to look at suspended particulate matter from deeper waters, for some reason that we don't understand, the ratio of 2 is always much, much higher than the ratio of 3. Either we've gained 2 or we've lost 3. You can't really tell which it is, but this ratio is massively skewed. And yet, these are real water column profiles from our data. This is from Hawaii, this is Hawaii surface water, and this is Hawaii mesopelagic water. And if you calculate the Tex 86 reconstructed temperature for both of these samples, they give the same answer. Because the loss of one is compensated by the gain of the other. And so you can maintain the same temperature across both things, but you have this huge change in signature of how these compounds are distributed. And Rich presented a really great talk about this at AGU last year, and also at Goldschmidt, I think. And you can go and you can data mine all the available data and see this for yourself. You can look at the GDGT 2 to 3 ratio and see that it has 2 patterns. One of which is that the sediment core top overlying water depth, as the water column gets deeper, this 2 to 3 ratio goes up. And so the deeper the core top is taken from, regardless of the sea surface temperature above, you're going to have more 2 to 3. Whereas if you're in a shallow water column, you're going to have 2 to 3 that's approaching 1. But the other thing is that it's a little hard to see here, but the warmer the sediment is, the more this ratio goes up. Or sorry, the warmer the sea surface temperature, or the warmer the local environment, the more this ratio goes up. And that's backwards from the predicted Tex 86 temperature effect. So the 2 to 3 ratio is increasing as sea surface temperature increases. That means you get more 2 as it gets hotter, but you're supposed to get more 3 as it gets hotter. And so this is backwards. And you can, again, plot this, and you see it goes in the wrong direction. And there are some other really interesting things that you can do if you look at the change in the individual GDGTs as a function of temperature. So GDGT 1 and 2 are sensitive to temperature, and GDGT 3 is not sensitive to temperature at all. It's just in there as part of the ratio. And so there's a lot more, I think, to be done here in terms of trying to understand where all this is coming from. But nonetheless, when you go and you look at sediments all look like this. Sediments have very little GDGT3, certainly not at the level of being more or less equal to GDGT2. And so most of the sediment composition here basically falls on this curve, and if you look in the water column overlying, it looks the same, and effectively this ratio is a function of how deep in the water column your sediment-water interface is, suggesting that what you're doing is you're integrating the whole water column locally. And so this points to the idea that, again, your GDGTs represent an integrated signal of the entire water column, integrated production flux minus some kind of a decay rate. So hopefully we'll be able to do more isotope work in the future to see if this is true or not. So I have tons and tons of backup slides for questions. I have at least as many backup slides as I have talk slides, so if you have questions, I have all kinds of other things that I would love to show you. But basically, hopefully, I've been able to convince you that when you combine isotopic measurements with lipid biomarker measurements, you can really gain insight to processes that are happening in the water column that are affecting our ability to understand sediment records. And the OAE2 example suggests that most of what was happening there was eukaryotic production, and the light values of delta N15 are a function of ammonia usage by eukaryotes rather than nitrogen fixation by cyanobacteria. The other part, GDGT lipids exported from the water column from archaea are consistent with a primarily subfluent water column source, and therefore we're still really up in the air in terms of understanding how these compounds correlate to sea surface temperature, and have ideas about that but no actual data. So with that, thank you very much, and I would love questions because there are gobs of slides I can show.
Any questions?
Well, I could ask the obvious, and not being in this field at all, but what turns on and turns off the eukaryotic production so quickly?
Well, it's not so quickly. So this event is estimated to take anywhere between 400,000 years and a million years, and there is probably some sort of a hiatus either before or after, and there must have been intense dissolution at the boundaries. So, I mean, the fact that the shale is overlain by instantaneous transition to limestone is a little bit misleading, I think. So, and there are chert layers, but there are also some sort of pseudo mudstone layers there. It's not uniformly 100%. It doesn't make any sense. 100%, 20% organic carbon in the shale. You can see that there is structure, and in fact, there has been a lot of Milankovitch modeling for what's actually happening there. And so you can see that there are intervals where it is more clayey and less organic, and there are intervals where it's more organic and less. So,
But it's still spoken, so the biostratigraphy demonstrates these hiatuses? Is that
no, I don't know. That gets beyond what I know about it. So the boundary is pretty sharp, but it's not instantaneous, I don't think.
Okay, sure, because there's something missing, not because something...
No, I don't know. I wouldn't want to speculate on that. I don't know the answer, but I know where I could point you to the answer in terms of other people's papers.
So what happens if you integrate the two talks? What about the GDDT at the OAE2?
Well, there's only, I think, one record, right? And that, I think, was actually OAE1 and not OAE2.
But are all OAEs created equal?
Don't know. We don't have a GDGT record for OAE2 to compare to OAE1.
From your nitrogen point of view?
I don't know. We don't have nitrogen for OAE1. So we have exclusive data sets. So no, I don't know. But there's certainly archaeal lipids are really tricky because there's a lot of discussion about how well they're preserved. And so there is a lot of archaeal lipid in OAE1, and there's presumably also a lot of archaeal lipid in OAE2. But there are a lot of old biomarkers in OAEs 1 and 2 because it's so organic rich. And a lot of what's done with biomarker measurements is to measure changes in concentration. And even if those concentrations are normalized to total organic concentration, that's still not necessarily an unbiasing type ratio. And the isotope values for the GDGTs in OAE1 are minus 18, minus 17 per mil, which is consistent with the DIC being 2 to 3 per mil heavier in those bodies of water, which, as far as I know, is true. That's exactly